the class. Okay, page 45. So, to be honest, you don't really need this demonstration to answer like any of the questions. There's no data table. It's like, I am just going to demonstrate the concept of chromatography and the connection between the plant pigments and then the questions that are all about the greenhouse gases is the idea that the more the plants photosynthesize, the more CO2 they take out of the atmosphere. It's just like this foundational assumption. And to photosynthesize, obviously, they need light. And different pigments absorb different parts of the spectrum. And we're used to thinking of plants as being just green, like one color, but there's two kinds of chlorophyll that absorb very specific parts of the visible spectrum. Let me at least pull up a visible spectrum. So here's the, Roy G. Biv that like everybody's used to.
I was afraid that had changed since I learned it.
What's that?
I was afraid that had changed since I learned it so long ago.
Oh.
There's so many new developments.
Oh, funny. Yeah, isn't that always the way? This is, down here, it's measured in wavelength. And I mean, we don't have to get like super into it, but the idea is the shorter the wavelength, the higher energy. So, and intuitively that the UV light is really high energy because, it's down here with the purple, and then lower energy down here. The waves get lower energy, so that makes them kind of lazy, I guess. So the plants are, again, very specific about what they absorb down here. And we're used to just thinking, or you're used to thinking of plants as being green. And the thing about color is that whatever color you're seeing is actually the light that the plant didn't want.
Right.
So the plants are absorbing this end of the spectrum and this end of the spectrum. And they hog that so that what we see is usually in here. And that's why a plant looks green to us. But even just to do that, there are, again, two different kinds of chlorophyll, which will look green to our eyes. And even within each of those types of chlorophyll, each chlorophyll type, it's chlorophyll A and chlorophyll B, they absorb two different, very specific wavelengths on the spectrum. So each type of pigment usually can absorb more than one type of light. And depending on the pigment that our eye sees, that's sort of a cue about which type of light they actually want. People are kind of more familiar with grow lights these days, and you might notice that they come in like sort of reddish and bluish, purplish lights. Because again, that's what the plant wants. The plant does not want the color that is coming back to our eye. So that's like the role of the pigment. And the more wavelengths a plant can absorb, and again, it's 2 for each type of pigment. And then each plant usually has more than one type of pigment. The more specific types of wavelengths that you can absorb, the more photosynthesis you can do. And so for us, that translates into ideally less greenhouse gas, less CO2. So I know it's nobody wants to do physics at 8:07 on Friday morning, but how is that? You get that for an overview?
Yeah.
Okay. Chromatography is a specific technique for working with pigments, and it pulls pigments apart, just separates them based on size. And again, I'm using chromatography paper that I swiped from the lab one of the last days that I was on campus. And it's, again, you can do this at home. It's just kind of like filter paper. So I also tried this out with coffee filters. Were probably the best substitute if you want to do any of this at home. Paper towels supposedly work. I suppose it just depends on the quality of the towel. My problem was when I used the paper towel, I'll show you, it did so much absorbing that it really didn't let the molecules separate out. Basically, the pores of the paper that are meant to trap particular sizes, it works on diffusion, and as we're going to put a little pigment on the on the bottom of the paper, stick this tip in a solution that will wick up and carry all of the pigments through, and the pigments separate out because the molecules that make up the pigments are different sizes. And it's like with like. It's like a little sorting system. Usually heavier things will settle out earlier because they can't get as far, and like smaller, lighter weight molecules. So sometimes you can see things that are hidden when they're all together. This is the story of the leaves in the fall losing their chlorophyll, and all of a sudden we say they turn colors. Color's always been there, the yellow, the brilliant reds, and the oranges. Those have always been there. For some reason, they're just, they don't have as strong of effect on our eye as the green hides them. I'll just say that. Like, it's sort of like all together it looks green, and then when the green stops, basically the wavelength of the sun is changing in the autumn. Literally, like what part of that visible spectrum we get in the autumn changes because our position is changing, and we are like a bubble kind of, and so where the light comes in to Earth's atmosphere, that'll change the which wavelengths we see. It just kind of shifts it more red. So we get different parts in the autumn, and the plant is responding. It's saying, hey, the green chlorophyll is not being as useful. Let me turn on my anthocyanins. That's the blue and the reds. Xanthophylls are the yellows, the oranges, and also some reds. So they've always been there. And something like, if I had a good tree leaf right now, I couldn't really find any. Most of the trees are kind of like flowering. But if I could take like a nice silver maple or a nice sugar maple leaf that was green, I could do this cartography example with that, and you would see what color it's going to turn and fall.
Oh.
If that sounds interesting.
Yeah, it does.
So your lab manual would have you use spinach, because spinach has at least has the two different kinds of chlorophyll, the A and the B, which are different sizes. So they settle out differently. And it's just, it's like green and other green. One's like slightly lighter and one's slightly darker. And then there's usually probably a xanthophyll, like something yellow. People are super familiar with carrots and carotene, beta carotene. that's a carotenoid, which is a type of xanthophyll, yellows. Oh, I don't know. I don't know if that helps or makes it seem weirder. So the way you do this is you, ideally you would make an extract, and I just happen to have a pestle and mortar at home. I don't expect that people at home would have that. But again, if you want to do this, I'm going to use basil. I had some basil, I had some kale, I think, which I'm going to use because it's got a nice purple. Should be some anthocyanins. Yeah, anthocyanin. Anthocyanin, as in cyano, as in every time your color ink goes out on your printer cartridge, it says cyano for that's your blues and your antho. Yeah, okay. What else do I have? So you could grab, I tried it with like this lemon tree behind me, but the leaves were too thick. You can do it with a house plant. I mean, you can try this with anything. So I'm going to do the extract. We're supposed to use sand, and I'm just going to use some salt. I picked up, this is actually kosher salt. This is a little grittier. And we want to, with the plant material, you want to, we're going to actually rip it up because we want to break down the cell walls, which are a little tough on a plant. You know, it's the thing that helps them defy gravity, that cell wall. So now I'm going to do everything I can to break it down so that the little molecules of pigment can come out into solution. And I can put that on the chromatography paper, and it hopefully will separate out some fun stuff. Some color. Put some basil in there because it's soft. I'm just, I'm ripping it up. Feels mushy. That's good. A lot of the cell contents are liquid. I mean, I'm literally trying to get the cytoplasm, you know, to goop out. I'm trying to break down organelles. That is my dog being thirsty. Sorry. They just sit active until I start talking to the camera, and then I feel like my animals wake up and have to be noisy. Okay. Pinch of salt. Okay. And then if we were in the lab, we use acetone. You may have acetone in your cupboards. That is nail polish remover. So it works really well to help chemically break down the cell wall. You don't need it though. Also, I have acetone. I actually had a little bit, but I couldn't use it because it was purple. Like the cosmetics industry had colored. It's just randomly purple to be pretty, I suppose. And because this is a color-based test, I want to put something purple. So I didn't use it. I just, for most of this, you could probably just use water. It is the universal solvent. But for most of this lab, for all our solutions, I found that just good old rubbing alcohol worked really well. I think we're supposed to measure, I'm supposed to put, I think 15 mils, but I'm kind of covering everything. It's really not that exact. I really, at this point, do want to separate solid from liquid. Again, if you don't have a mortar and a pestle, you can do this at home. With it, kind of like any wooden handled thing. I have a muddler. This end of a wooden spoon. just in the bottom of a cup, even a blunt end of like a butter knife. And you know, you really, again, you're just going after the organelles that hold onto the pigment molecules. And we want to break those organelles down. So I got to get into the cell wall. Anything that'll rip and tear. And the salt is helping be abrasive. See, it's already getting pretty green. Man, you could even rip off a little piece of sandpaper and like attach it to the end of a wooden spoon or something. It is nice and dark green now. Okay, so at this point, I just want to transfer it to the paper. Get a few things ready. We're supposed to use a capillary tube. So anyone who's ever used a straw to like drop water or hold it so you can, I don't know, flick it at somebody. Same concept. We're using basically water surface tension and adhesion properties to like climb the wall. You probably don't have a capillary tube sitting around at home. That's okay. I actually didn't, I thought I'd find something a little better than this, but I just happened to grab in these transfer pipettes. So I'm just going to use that, but even a coffee stirrer would work for this job. And just the narrower, smaller diameter, the better. Again, I'm going to skim it off the top. Yeah, I don't need much. I just need a drop. I don't want the solids. And then I'm just gonna, guess it's gonna be a little absorbent and I want a small spot. Not so good. I gotta get it off this paper. I should just keep going. I should let it dry. Just takes a few minutes and I should just keep piling on. I'm a little off center there. Oh, ideally I should filter this to separate out the solids, but it's not that big of a deal. It'll still work. Yeah, just completely graceless there, and it'll still work. Okay, so the solution, the chromatography solution, I'm basically just going to do like half and half tap water. We have a well, but just tap water. It doesn't have to be DI water even. And that rubbing alcohol. It's kind of eyeballing it, half and half. I mean, again, you don't even have to measure. I want to say your manual says something like a half an inch of water, which is a little bit of a rough measurement, but I can't remember. I think you're doing that in a test tube, so it's a different, yeah, it's a different diameter. I just want it deep enough that I can get that tip of the paper in and not much else. So while this is kind of drying, I'm gonna get it ready to hang, and I just, instead of, let's see, your manual has you use a paper clip and a rubber stopper. I just use this pencil and some tape. I just kind of eyeball it. Not the big deal. It's stuck on my finger now. I need more tape. Try that again. I think that'll hang low enough or close enough that I can adjust it. And then I'm just gonna get some tape off because I want to be able to stop the pencil from, I want to leave it. This can take up to 30 minutes to let the solution carry the pigments all the way through, so I don't want to have to sit here and hold it. So I've just got the tip of the chromatography paper in there. Just crudely stopping the pencil. Sometimes it gets annoying. You don't want it to touch the sides. And this is pretty pale, but at this point you can kind of watch the solution just carry the pigment up. We call it the dye front. You can kind of watch the dye front move up the chromatography paper. Again, you can just take a coffee filter and cut it into this kind of shape. It doesn't even have to be that pointy shape at the end. The pointy shape just helps you concentrate where the solution is coming through. Sometimes you'll see in a minute how you want the smallest point of contact between the paper and the solution because you don't want the pigment to leach back into the chromatography solution. And again, you just kind of sit and wait. And in the end, put that to the side and let it do its thing. In the end, I got this. We could see there was some darker brown pigments and then a lighter green. And then this darker green, out sort of at the farthest rims. Ignore the blue stuff. This stuff and this stuff, that's stains from another thing. So that was like the best I got yesterday without being able to use the chromatography solution. That probably would have helped a little bit because that is made of acetone and ether. So a little different. But the isopropanol works pretty well. I also tried some things, some other household items that are a little bit more interesting to use at home is ink. Ink always has hidden pigments in it, particularly black. This is a felt tip marker, just Sharpie, ballpoint. You know, I just make my line, and I do the same thing. I'm just gonna roll it up on a pencil and hang it. And this one will be much more dramatic than the plant thing. So paper chromatography is specifically for doing this with pigments. Again, like anything. I also messed around with food coloring because your non-primary colors have the hidden pigments, right? So I used green food coloring, but guess what pigments it pulled out?
Blue,
And
yellow.
yes,
Oh yeah.
I'll show you.
Oh wow. That's cool.
Yeah. A little more exciting than the basil. Again, a tree leaf, doing this in the summer. And this is, I don't know, maybe you're bored. Maybe you're at a family reunion and there's some kids to entertain. Seriously, a coffee filter and a tree leaf, and this will happen. It's really funny. Let's tape this up. But the pen ink, I think you'll really like. There we go. You see that line okay?
Yeah, the ink is not submerged, right?
Right. Yeah, that's ideal. Is to keep the pigment. I just did a lousy job being tidy, but oh, even our plant. Ooh, oh, I didn't do a bad job. Hey now.
Oh wow.
It always kind of reminds me of like chalk on the sidewalk that gets wet. It's very poetic. I'll probably put some photos of these things on Instagram later, just for fun, because they're pretty. Here's the plant, or sorry, the green food coloring. You can kind of see the difference between the chromatography paper and the coffee filter. It's a similar effect. It just doesn't separate as much. It's just like the green is a tighter band, and the blue didn't come out quite as much. But maybe if I'd have let it go. I was up doing this until about, I think, between 7 and 8 p.m. And I was like, oh, I gotta call it quits. This is getting silly.
So that is just a function of the weave of the paper between the chromatography paper and paper filter,
Yeah,
coffee filter.
right,
Okay, I get it.
right. The paper, right, like the matrix. Yeah, there's pores. And
Okay,
filter paper is meant to trap some and let some go through,
all right.
right? So I don't know. There's probably like a size, a pore size that's different between the chromatography paper and the coffee filter. I imagine this one's finer.
Yeah, makes sense.
Coffee's just meant to separate those giant coffee grounds from the water, the liquid part. So, well, you can kind of see our pen ink is doing what it does right away.
Mm.
Start getting the warm tones. Warm tones always kind of settle out first, it seems like. Oh yeah, and this can take up to 30 minutes. I can show you an end result. What did I just do? Pen ink. There's 30 minutes.
Oh, wow.
Yep, all of that is in the black marker. And again, any black, because black is all the colors together.
So why, when you mix the Easter egg dyes all together, it just comes out like a gross greenish brown instead of black?
Um, that's a really good question. It's about how much of which colors you put in. And if you're getting the brownish, greenish, that means you got too many, it's too even. So what you have is complementary colors that kind of cancel each other out to get that, usually grays or browns. Sometimes the
Oh,
grays can look greenish. You need more dominance of something, either because blacks can be either warm or cool depending on which colors are. But again, you don't want opposites in there in equal balance because they'll cancel each other out. So you don't put purple and orange. We'll do that. Red and green.
oh.
We'll do that. You know what's funny is as I say that, those are also the two areas that color deficient people struggle in. So they don't see the red on the traffic light, they see a brown, or they don't see the green, they see two different shades of brown or gray. It's funny. Just put that together. It's complementary colors. When people talk about color theory, this is the kind of thing you're talking about. You're manipulating how the eye works with the electromagnetic spectrum. And some colors are just, we're meant to see more green. Well, that phrasing was really non-scientific. Our eye picks up more green than any other. It's like, usually it's the fattest area on the visible spectrum. There's more shades of green that we detect or we're capable of detecting. And like some animals are different. Yes, insects tend to see UV stuff that we can't see, that's off our visible spectrum. Bees, bumblebees and honeybees see what we see. We have they can see UV light, but they can also see like the same colors. The different insects are attracted to different colors. They'll dress like a flower, you know, because they can see yellow, they can see pink. I mean, notice there aren't a lot of really true blue flowers out there. Usually a florist has to dye a carnation to be that powder blue. It's because flowers, since they're meant to attract pollinators, they tend to be in colors that insects can see. And it's weird that we have like exactly the same spectrum as bees, except that they also have UV light. So maybe I should show you. This is kind of cool. So they can also see things that we can't see on flowers. These things called nectar guides that are meant to be seen in the UV spectrum. So to us, the flower looks like this. And to the bee, it looks like that. It's supposedly to guide them to where the pollen is. So for a plant, it's just pigment is everything. So that's why they connected the chromatography, which is specifically for pigments, with all these greenhouse gas questions. They just sort of forgot to tell you. I think when I read it, I was like, I just don't know why. There's no data table or anything. So normally when you do chromatography, you can calculate molecular weight based on where stuff separates out. I can get a little bit more technical. So let's see, what else did I do? Anything interesting? I guess yellow and purple. I used, again, food coloring because I knew it would have some hidden pigments. Even yellow in food coloring has a darker sort of orange, and then the lighter yellow. So I got that to come out pretty good. And then this was purple. So this was me. I didn't have purple. I made it according to the box, which is basically like double the amount of red. Because so I put in like 4 drops of red and 2 of blue. And I just put that straight into my chromatography solution. And it separated. It's pretty good. When I did it with coffee filter and paper towel, not so much. Yeah, so some things work really well. This probably would have worked, the coffee filter, if I'd have just left it longer. Oh, and yeah. So that's the gist of paper chromatography. You might also hear people talk about gas chromatography, and that's not for pigments. That's for when we want, like, if you have a solution or people, I don't know. I was going to try to do this with essential oils to see how diluted they were or how true they were to what they claim to be. You can take molecules, and again, the gas chromatography separates it out kind of like this, but it does it in the air. And it can measure the gases, much like oil refining. Molecules will just sort out by size under certain pressures and temperatures. And the gas, you put it in a column, and the gas chromatography can tell you, hey, you've got 80% whatever lemongrass, 10% water. Like, it can kind of tell you what's in there. So it's the same kind of thing, like identifying molecules that are kind of hidden to our senses and separating them out. Anything you want to look over the questions while you have me here to ask anything about how to answer those?
Like identify the greenhouse gases in the photosynthesis reaction.
Mm-hmm. So they give you that reaction on the prior page, 45.
Yeah, so carbon dioxide.
So where does the plant get that?
From the environment, from the air.
Atmosphere. Yeah, they literally, plants have pores like our skin, and they breathe it in.
So that's the old theory of, if you talk to your plants, they thrive because they're directly getting a lot of CO2 from your exhale.
Yep.
Yep.
Stinging. Mm-hmm. 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 On a day like today, they're balancing that. They're like, hey, I want to open up for air, but there's not a lot of sunlight. So I'm limited in what photosynthesis I can do. So I'm not going to open my pores all the way. They kind of open them like halfway. And when the sun comes out, then they'll be like, hey, let's look. let's do the photosynthesis. But right now, because every time they open up to get CO2, they lose water. It's not a good thing. It's just an inefficiency in nature. So they kind of balance it. So anyway, I always joke that that's why you feel tired on a day like this where the skies are gray. It's just, because plants were on Earth first, it's just sort of how all life developed. When the sun's not out, we're just like, there's no point in being awake. I can't get my sugar.
Yeah.
So
So
it's a little gift from me to you.
that's pretty cool. In the fall, when leaves are yellow but not crunchy and crispy, they're still doing some photosynthesis
Mm-hmm.
through the pigments that's still in the plant, but the green has died off
Yeah,
or
because
gone away.
so I forget which is which, but again, chlorophyll A absorbs 2 different wavelengths and chlorophyll B does 2 different wavelengths. I forget who's who and which is which. But at the time of fall, because we are turning away from the sun, again, the part of the visible spectrum is shifting and we're not getting as much of the red and blue that the chlorophyll absorbs. We're starting to get more of the middle. It sort of shifts. So the plant has to change. It knows it's going to get more light if it, can keep up with the changing wavelengths. So it's like chlorophyll just becomes ineffective or less effective. So why make it? Go ahead and maximize whatever wavelengths it is that the xanthophyll absorbs, which is probably two different. Most of the pigments seem to have these 2 different, they call them photosystems that do the job of absorbing, and they each can only do 1 wavelength. I mean, it's not magic. It's pretty, it's machinery. So, and if a plant turns more like a purple, gosh, like there's that one kind of maple that I think it's called a maroon variety of Norway maple. It's so dark purple. It's like plum or wine or burgundy, that's absorbing a whole different set of wavelengths compared to the more yellow orangey.
Oh, yeah.
And usually whatever it looks like to our eye, that's the wavelength it doesn't want. So you gotta it's always opposite. It's always freaking opposite of what you want it to be. That's with light and our eyes. I'm like, what do I want it to be? And then flip it. Oh yeah, that's right. That's the right answer. Because it's light. And the plants have, been doing this for 13 billion years ish, something like that. I mean, it's like a long time to perfect their game, So they're very sophisticated about. And again, the whole time that they're green, the yellows and the purples, they're still also doing their job. It's just that towards autumn, the chlorophyll again becomes more, I should say, just less useful. So you just drop it. But they're all usually there kicking all the time, because even during the day, as the sun moves across the sky, the different parts of the spectrum, it changes. So the plant's just there all day being like, okay, let me get that morning light. Let me get that afternoon light. Let me get that evening light. Because the more sugar, the better, the more you can pack away before winter. I mean, it's, and they don't have to go to work. They don't have to do homework. This is all they do. They're just sitting there all day, hogging all the light. And it's why it's shady. You got a tree that's got a lot of big leaves, dark green leaves that, overlap each other, a dense canopy, and they don't leave any light. They're literally the pigments are absorbing that light and not letting it transmit through. So it's just controls everything of photosynthesis. I could talk about this all day. I love it. It gets very complicated the more you get into school and go with the plant physiology, becomes more biochemistry after a while because they have it nailed down. They really, the details of photosynthesis could be a whole semester course. It's so wacky, and there are some parts of it we still don't know. We have a couple of black boxes. There's a part of the story they sort of hypothesize a couple of organs, but they're not sure, or organelles. Not totally sure. I'm not totally sure how plants sense gravity. So they hypothesize that there's a molecule to do. It's it's crazy. Exciting world of plants. So for that first question, you just use that reaction. The greenhouse gases are the carbon dioxide, and then I guess the water counts since water vapor, but
Tough to figure out where the sunlight comes from.
I wrote the sun. Fusion.
Yeah.
You could be a real smart mouth. The water, I just put the hydrological cycle. I mean, it comes from, the plant can get it from the soil, it can get it from the air. It can collect it through the roots and the leaves. Some plants prefer to get it on the leaves. Some would prefer to never have it on the leaves. It's just, I think the rest of this is just greenhouse gas stuff. Sunlight quantity use. Well, so number 2, if the plant doesn't absorb it, it would pass through or reflect back. And that could ultimately affect the temperature in the atmosphere. So if you're talking about greenhouse effect, you're talking about warmer temperatures. Less plants would mean less light being absorbed. And the other thing is, in light of what we've talked about with pigment, the more biodiversity of plants you have, the more light would be absorbed. Less would reflect back up to the atmosphere to heat things up.
So theoretically, on a huge scale, photovoltaic solar cells could hurt the environment because they would absorb more light if they were like everywhere, and there wouldn't be enough light energy for plants to survive. I mean, on a crazy Mad Max type scale. You buy me?
Yeah, I mean, it would take a lot of solar panels.
Yeah.
Really, it's I mean, your point, I feel like more applicable is like hey, if you're in a really densely canopied environment, maybe solar panels might be tough because you got to get them, the plants are going to hog them from the solar panel. Maybe that's the flip side of what, but yeah, you're right. There's a finite amount of light. I think it's a pretty good amount. I mean,
I just remember hearing a congressman say something like solar energy is not good because it would take all the sunlight from the plants, and I thought it was so ridiculous to hear that. But
yeah, that's
actually on a really overthinking type way.
On that scale, all the carbon monoxide that comes out of inefficient cars are gonna kill us, smother us.
Yeah, right.
And it's like, well, if you're in a box, I guess, but that's why you don't run it in the garage with the door closed, right? Like,
yeah.
I get it. It does, with all the hydroponics, it does sort of get into like, well, if this plant, you don't want plants to compete for the spectrum, the parts of the spectrum that they need. So you might grow like a more purpley colored kale so that it's competing less with your green spinach. If you're coming from a grow light, when the sun, I wouldn't worry about competing for light too much. I mean, you worry, it's really hard to grow grass under Norway maple trees because those are the ones with the really dark green leaves and dense canopies. You can almost ID a Norway maple because the grass is really sparse. Just not, they do compete, but that's a little system. I don't think it would, happen for the solar panels and stuff.
Yeah.
But when you have grow lights, you have a very finite source of photons. And so we do think about that in indoor growing or small space grow, anything with grow lights, they will compete. And you can pick different pigmented plants knowing that they favor, it's niche partitioning.
So do grow lights come in different wavelengths?
Mm-hmm.
Would make sense.
And the cheapest and the easiest is just to get a white light. That's what we had, in the green, the lab on our grow shelf. Those bulbs, those were just full white spectrum. So that's all the spectrum. But you can get more sophisticated. I don't know if this is still going on. I feel like the testing was sort of in process. But when LED lights came out, a lot of people switched to LED lights for growing indoors and for hydroponics, and they will come in red and blues because why waste your, when you grow stuff indoors, it's super energy intensive. So you got to run those lights as compared to outdoors.
Yeah. And
why pay for that wattage and run a part of the spectrum your product is not using? So you don't want that. Why waste it on that middle yellow green area? So they come in the blues and the yellows. So I'm going to pick on Loyola again, that lab where those students process the waste oil to run the buses. You can see from a distance because most of that building is it's geothermal, and it's also a lot of passive solar because, again, they're like a block away from Lake Michigan, so they get a lot of sunlight, and it's a lot of glass. So just like even riding on the train, if you go by at night, you'll see this purple glow coming out of that building, and that's their grow room where they just have plant science classes, and they have grow lights that are I think LED, and then but they're blue and red. That's my point. So from a distance, it looks purple. It's just a coincidence. And then you can buy like just the blue or just the red, or you can actually control the rate of photosynthesis, with plants. You can, by putting different lights on them.
Makes sense.
They'll like kick out all these oxygen bubbles, and they'll do it faster if you put on the red or blue light. If you give them a green light bulb, all the bubbles slow down. It's just, I don't know, it's very machine-like. Any other of these questions? You don't have to do number 8 because you're not, I mean, if you want to try some chromatography, you can attach your strip. Yeah, most of this is sort of connecting the more plants, the less greenhouse gas, less temperature, less global warming. So I think that'll covers everything. But anything I missed?
Nope.
Okay. Well, I'm gonna dismantle landfills today, so I'll get that data up on the table, and then I'm gonna weigh all whatever's left of the trash, what I can find. I'll just pull everything out and take videos. So, because I figure you guys will be able to identify things better than I can. Like you probably wrote down the different pieces of trash. But I'll see what's there and try to sort it out. If I have any questions, I'll email or leave a comment or something, prompt you guys somehow. But other than that, I'm just gonna weigh every piece, and I'll add a column to that data table for the decomposition weight. And then you guys will subtract it, should be less than the original weight, or at least the same. Somebody did an aluminum can, and I'm guessing that might be, the weight might be a little close, but hopefully nothing gained weight. I suppose something could have absorbed water, but the idea is that the decomposition should have like eaten away. And if it didn't eat away, that sort of sets you up for what we're going to talk about next week with waste management and the different types of materials and how they break down or not, and how we try to recover what we can. And I think the quizzes are up and ready to go. People want to work on that this weekend. And then we have one more of the Zoom sessions on Tuesday. And then that's it, except for the final.
Really?
Yeah.
Oh. Okay.
Yep. Lickety split.
I've got to get some of these labs turned in then.
Yeah.
Okay.
Grades are due, I want to say April 30th. So you kind of got like all of next week. If people want to turn things in, people need until like Friday, Saturday, Sunday. That's fine.
Okay.
Cool. Well, let me know if you have any questions. Try some chromatography. What else are you doing? It's Friday night.
Oh yeah, big chromatography night. Well, we're still under boil advisory. All the construction in the north end, our
Oh,
water was turned off yesterday. We didn't know it, so it came back on last night. So we're under boil advisory for another 24 hours. So I couldn't even make coffee except for bottled water I had stashed away. So
thank God.
yeah, it's just great.
Oh, yeah.
Stay at
That's
home
an
and
awful
no
sign.
water. So, all right. Well, have a good weekend.
You too.
See ya.
Bye.
Bye.